look at the data, there are 9 patients, so that's about 15 patients, which have sustained high antibody titers in the LOTS clinical trial. And these titers have ranged from 12,800 to as high as 819,200. And this particular patient with the very high titers, as you can see from this publication, did have a clinical decline also. So I think when the titers are extremely high and sustained in a late onset patient, it becomes more apparent clinically. But I'm starting to question what happens to those patients who have those persistent high titers but may not be as high as 819,000. So that's work still in progress. So the next part is, what do we do about this, and do we have a way to study it? So just as a quick reference to this, I want us all to understand, because we've talked about antibodies over the last 2 days, and so I'm going to try and do a schematic of this. This is the therapeutic protein, which is the enzyme, and this goes to what we call an antigen-presenting cell in the body, which then breaks this protein down into smaller units, which we call peptides. And then this is presented to cells in the body called the T helper cells, which works with another cell called the B cell, just to keep this simple, which then makes the memory B cells. So this is the memory storage of the body that I've been exposed to this antigen. And then the B cells also make what's called a plasma cell. And then the plasma cell is the ultimate source of antibody production. So I want us to just look at the schematic, because as we talk about drug therapy, we'll see why we use what we use. So the clinical settings are as follows. We've now identified someone who's CRM negative, who's someone at risk, and so we can start them early before they've been exposed to the enzyme. The second is, after exposure to the enzyme, they've started to make high titers, or they've started to have a very quick response to antibodies, to the antigen. And then the third is, we've already made very high sustained antibody titers. I think there are 3 completely different challenges in the field and complete different ways to manage them. So in the naive setting, if you can think about it, if we preempt the body before the exposure, it's a much easier task. And so these are agents which we don't have to go into the details of them, other than to understand that we've used these based on experience from other disease conditions. And so it's an application of these agents now to our individuals with Pompe disease. And this is, of course, work which was done in collaboration with Genzyme and with many, many people across the globe. And so if you look at these agents, where do they act? This is where the drug methotrexate acts. It acts at the level of the T cells and the B cells. And then you look at rituximab, it's acting at the level of the B cells and the memory B cells. So remember, I talked about 2 agents, but look at the entire pathway and let's see if this works. So this was our first report in the New England Journal in collaboration with Nancy Mendelsohn. And this was the first breakthrough that when you provided these agents to someone who was CRM negative, who had been exposed to enzyme and had made antibody titers, that we are able to bring those antibody titers very successfully. Now, how is this patient particularly doing? Do we continue to give them these immune suppressive agents, which are not very safe in the long term? And I just want you to look at this line in red, which tells you B cell recovery. This is a simplistic way of telling you. This is showing you B cell recovery, and the blue is the antibody titers. So you can see now with this patient of all immune therapy and continuing to receive the enzyme, has completely what we now say a state of being immune tolerant. So the B cells are doing its job, and the antibodies are not coming back to the enzyme. So this is a second case. Once again, concentrate on the red line and the blue line, and you can see another situation of complete recovery. This has been after exposure to the enzyme. They've already mounted an immune response, and we've still been able to successfully tolerize them. Now comes the ideal setting. I've identified a CRM negative. The doctors called me up. We've been on the phone, and we've now gone in before starting enzyme replacement therapy, or done it at the time of enzyme replacement therapy. You can see that the bars on top is just very short course of these agents, over 9 weeks. And within a short period of time, we've been able to cause success with a downward trend and tolerization of these patients, and a recovery, because you're seeing the red line, which is the CD19. Now look at the next case, patient number 4, a similar story. And so we've got follow-up of these cases, and these have all been 4 CRIM-negative cases, and this paper is currently in press in Genetics in Medicine. I must tell you, this was a hard one to publish because this is new emerging data, and it's often looked as kooky science, but this has shown success, and I'm hoping that with more of us in the field, we're going to be able to do this more. So how are these children doing? If I told you earlier that the median age of death of a CRIM-negative baby was 27 months, we've, I think, gone right past this, and now we've got at least 15 more cases which are being treated with rituximab, methotrexate, and IVIG throughout the world, and we're on the phone as a team trying to help out. And all these 4 cases, as I told you, are completely tolerant. They're able to get immunizations, they're able to get the enzyme therapy, and they are continuing to show progress. So this is the first little one who was started on enzyme therapy. And so this is case number 1. So you can see he's on a ventilator at this time. This is him. He's now coming off the ventilator for several hours in the day. He's making motor gains. This is patient number 2. So she's walking, so this is quite unique. I'm just praying that I do not have a computer crash today. Okay, and now this is the current status, the first cohort of our long-term survivors of CRIM-negative. And if these videos work, that's great. If not, I'm okay. So this is on the left. This is the little boy who's now over 5 years of age. The middle one is 5 years of age. And this one is a baby from Israel who's also 5 years of age. So we've got the first emergent cohort of these CRIM-negative cases who were treated with immune modulation, who I'm hoping, but if these, I'm fine with this. It's fine. Don't work. So these children are actually walking and doing quite well. So now our next challenge, as I told you, is this clinical scenario of high sustained antibody titers. I think this is a real challenge in the field. And if you look at it, we had attempted with our first 2 cases of antibody titers, the first 2 cases at Duke, we had completely failed, and these children both succumbed to the disease, and this is how you can see it. This is one of the 2 cases that was treated with plasmapheresis, cyclophosphamide, and IVIG, and you can see it did no different despite all these agents that were used, and succumbed to the disease. And so this is looking at the literature of the various agents that have been used, and we've not had any success to date. And so I want you to look at this slide one more time, which I showed you. This is an area which I think we've all overlooked as a scientific community, the plasma cells, which is the final source of antibody production. So I showed you where the methotrexate and rituximab or other agents can work. They're acting at T cells and B cells, but we have nothing to date which is working at the level of the plasma cells. And so why target the plasma cells is because they're the final source of antigen-specific antibody production. Each plasma cell in the body makes several thousands of antibodies, not per day, not per week, it's per second. And they can survive days, they can survive weeks and months. So they're very non-forgiving and they have very good memory. So there's no success in eliminating these high sustained antibody titers with the agents that we've discussed to date. But then we looked at the literature, and there's a drug which has been used in multiple myeloma, which is called bortezomib, which acts on these antibody-producing plasma cells. And so how bortezomib acts is, to keep it simple, is that it acts as a proteasome inhibitor, which is present in large numbers in these plasma cells. And so if you destroy the plasma cells by giving bortezomib, you will not make new antibodies. And so using this strategy, we had the good fortune of the trust of a family who had a baby with Pompe disease, who was 4 years old at the time. And this patient, this little girl, or this little boy, was started on therapy at 5.4 months of age. The clinical course on ERT, so as expected in the first 6 months, did extremely well. The left ventricular mass, or the heart, improved from 260 grams to about 135 grams. The baby was making nice motor gains. And then the antibodies were being tracked, and you can see that the titers are going up. And as the antibody titers were going up, the baby started to have a clinical decline, started losing motor milestones, now started requiring ventilator support, required G-tube placement for feeding, and the left ventricular mass started going up from 135 to 360 grams per meter squared. And this one, I wish these videos would work, but you can see this increase in the left ventricular mass. As you can appreciate here, all of this is left ventricular wall thickness as compared to here. And then this is how the baby looked after the first trials with cyclophosphamide and rituximab. And then we started with bortezomib, and in the interest of time, I can just show you that with 2 or 3 rounds of bortezomib, we were able to bring the titers down to 1 in 400. The left ventricular mass came down from a high of 360 down to 85 grams per meter squared. And you can appreciate this here. From having all this obstruction and not being able to really function well from a cardiac status, this child really was able to start coming off the ventilator, the heart had improved, and there was a significant recovery. So these are the changes. There was a decrease in vent settings. No side effects were noted on this. Case number 2, so was this fluke? So a similar story, very high titers, 819,200. This particular baby now has a titer which has come down to 12,800. The left ventricular mass index has come back to the normal range, similar response. And so we have now started this, and we have several additional patients on this regimen, and we are hoping to bring this information to light. So in summary, I would say high sustained antibody titers is a poor prognostic factor. It's important to monitor these antibodies periodically. It's important to predict patients at risk. There is a need for immune modulation. That we have had some success, but there's a lot more work that needs to be done. Gratitude to this first family that trusted us with the bortezomib work. This is all my Duke collaborators. My acknowledgements to all the treating physicians around the world, and this number has really grown. I've not really updated this. And I think about this because Maurice is in the room here, and just like the adults where her mother says, my daughter has Pompe disease, and I thought I would lose her early, but we have hope. She's ready to fly. I think that even for some of these infants and some of our individuals on enzyme replacement therapy as adults, I think we are making new strides in the field collectively. Thank you.
A completely different subject. In fact, Tiffany asked me to give a lecture on muscle regeneration, and I said yes, but it is a very complicated story, and but all in all, I thought it was very good that you learned something about how the muscles work in our body. And what I would like to address, as mentioned, it's a complicated story because tons and tons of literature have been written on all the subjects that I've listed here. But what I would like to address today is how do the muscles develop in the humans? How do muscles grow from infancy to adulthood? How are the muscles composed and how do they function? And what is the effect on glycogen storage? And how can muscle damage be repaired and what mechanisms are involved? And at the end of my talk, I would like to give a short introduction to muscle stem cell therapy experiments that are done in our Stem Cell Institute at Erasmus University. So development of the muscles in the humans. And I start with something that Arnold already touched upon yesterday, genetics. If we're born, we receive half of the genetic information from our mother and our father, and all this genetic information is laid down in the chromosomes. And when you become a girl, then you have XX, and if you become a man, boy, you're XY. All this genetic information is included in the DNA, which consists of more than 3,000 million base pairs, as Arnold already mentioned yesterday, and consists of more than 25,000 genes. And in these genes, and this is Roger Federer, you know already from start, maybe whether you will be good in sports and whether you will win Wimbledon or the US Open. But also in this information, it contains the structure or the code of alpha-glucosidase. So is there a mistake in the alpha-glucosidase gene? Is there a fault? So there already is also laid down already that how are your muscles composed and what are you becoming an athlete or not? So everything starts with the fertilized egg, and these fertilized eggs divide and become more cells, and at the end you get the human body, and this human body is composed of all types of cells and a tremendous amount of muscles, and all different organs like the heart, the brain, the liver, the spleen, and as mentioned, the muscles. And now I would like to go back to the beginning. What you see here is an embryo of a chicken at day 2 or 3, and that's comparable with the human embryo at day 27. And what you see in this embryo already is that, for example, there is the heart already starting to develop. There is the eye and the otic vesicle, so the ear that is going to develop. And what you also see here is that the forelimb, and in the chicken that's of course the wings, are going to develop. And at day 3 you see it's already more developing, and the hind limbs. And if you take a cross-section at the side of the forelimb, and we go to the next slide, then you see a transverse section of this little chicken, and this is a human embryo. Then there is in the middle the neural crest, and next to it, where the red circle is, there is an area, and it's called the dermatomyotome. And in this dermatomyotome, in the myotome, there the muscles are formed. And these muscles are then migrating to the limbs and to the chest. And so the muscles are formed from the center and migrating to your limbs and the rest of the body, each on the different levels of the somites. And as you can also see here, and this is a nice picture, the purple are the migrating muscle progenitor cells. And what you have to realize that during these first 3 to 5 weeks of the development of the human embryo, already the muscle progenitor cells are formed. So there, the content and the future of your whole muscle composition is already determined. And it is even so that if cells do not develop a certain marker during these first 21 days of development, then muscles can't differentiate later in life. So it's very important. Now I would like to go to the human body. And here you see a little premature baby of 24 weeks old, and such a premature baby is about 600 grams. And until the baby is born, so term age, 40 weeks, so in 16 weeks' time, this baby multiplies his weight with 5 times. So an enormous increase of weight. And then, if you become a man, an adult man, again, the weight increases from the neonate to adult size 25 times, and from a premature to an adult man 125 times. And if we look at the body composition or the muscle composition, the muscle mass even increases more. 15% of the human body is muscle in a premature or term baby, and when you're an adult, it's 40%. And if you look what that means to the muscle mass, then the muscle mass increased 300 times from the premature baby to the adult male, and 60 times from the neonate to the adult male. We can do something about our muscle mass, so we can train. And what you heard yesterday, then the muscle fibers increase, and also there can be more muscle fibers. So, in fact, this is exactly the target of our muscle training program. But at the end, how much you train, you will lose muscle mass at the end. And with aging, the muscle mass decreases, and this is a process that is called sarcopenia. So then I would like to go to the muscle. How is the muscle composed? And here you see a shoulder muscle, so the shoulder area. And what you see here is the junction of the muscle to the shoulder bones, and that is called the myotendous junction. And here you see the muscle mass, that's the muscle, this is the biceps muscle. And this muscle is composed of a bundle of muscle fibers, and that are all these round circles in the middle. I think it's clear. This one and that one and that one. There are bundles of muscle fibers. And you see also one here, a myofiber. You see that this myofiber contains all little parts, and that are myofibrils. And a myofiber is the cell. And if you see this, you see, I don't know, oh here. For example, this blue little nucleus, that is a nucleus of the muscle cell. And here, a little bit higher, you see the satellite cell. And I will come to that later. But first, I would like to go back to the myofibril. The myofibril, that are the contractile elements of each muscle cell. And this is a myofibril, the contractile elements. And this myofibril consists of actin, that are the thin filaments, and myosin, that are the thick filaments. And when a muscle contracts, these filaments slide across each other. So then the muscle, these myosin and actin filaments go inside each other, and the muscle contracts. But what you can also see here is that there is desmin, and desmin is part of the cytoskeleton. So the myosin and the actin has to be attached to a cytoskeleton. And you have titin, for example. And it's also part of the cytoskeleton. And I would like to show you that here you see the C line, and here where you only have myosin, it's the M line and the Z line. So, and that's between C line, you have two Z lines, you have the contractile element, one contractile element. And so that's important for the next slide, because here you see skeletal muscle of a healthy individual and of a patient with Pompe's disease. And in fact, Nina Rawan also showed something like this, and maybe I can go back to that. You see here the Z line, and here in the section of the patient with Pompe's disease. And between the myofibrils, you see the mitochondria here. You see them also here in the patient with Pompe's disease. And then also between the myofibrils, you see a lysosome filled with glycogen. And what is now important, and I think this very much touches to what you just mentioned about the autophagosomes, is that these glycogen-filled lysosomes are between the myofibrils. And when these myofibrils want to slide over each other, then you see that there is the lining up has been disturbed. And can you imagine that only already by this process, that the glycogen-filled lysosomes are between those myofibrils, you lose strength already. And in fact, that was also shown by Maarten Drost. And Maarten Drost is from the faculty of Maastricht. And he showed that, for example, in mice with Pompe's disease, there is an increased content of desmin and titin, and probably because there is more strength on this cytoskeleton. Is that still clear? Okay. So, then we go to the glycogen storage in skeletal muscle in Pompe's disease. On the right side, you see a cross-section of skeletal muscle, and these little red dots are lysosomes, and this is a patient with only very mild glycogen storage, opposed to what you see on the left side, where there is severe glycogen storage and destruction of the myocells, of the cells. And if we go to the destruction process in infants and adults, then you see in the top panel the destruction process in infants, and on the left you see only a little bit of glycogen storage, and that increases here. All the muscles are damaged, and in the adults it's a much more variable process, where here you see a little bit of storage. The purple is glycogen, and here you see that there are fibers that are completely destroyed, but there are also fibers that are relatively normal, and that's also what you can see here. So the process is much more variable and also much slower. So I don't want to go in too much detail, but with enzyme therapy, the enzyme goes in and to the lysosome finally. I will skip this one because Nina talked about it. And if we see what happens, can we restore the glycogen storage, then at least it's a complex story. And I don't want to talk about autophagosomes, but at least we can say that something happens in the muscle. And if you look at the top panel, you see that the glycogen disappears in this patient, while here the muscle cells are beyond repair. And what is also shown here, this is a complex slide, here a normal cell, and what I want to show here is green fibers are type 1 fibers, red are type 2 fibers, and here you see a patient at start of therapy. These are the same panels, and that also in these patients with infantile pompous disease, both the red and the green fibers show some response to therapy, and here you see that the muscle cells look much better than there. So something happens in the muscles over there. And then,
which? The green and red, I'm sorry.
the green. The green is type 2 and 1, and the red is type 1. Yeah? But maybe it goes, yeah, I want to show it longer, but that's not the point I would like to address. I would like to go further to what happens with muscle damage, and what are the capacities of the muscles to repair. And here again you see a muscle fiber, here the muscle fibrils that are in the muscle fiber, the myonucleus, but what's important here is that there are also satellite cells. And satellite cells are outside the muscle fibers, but these satellite cells are a kind of resting cells, and these satellite cells has the capacity to divide at the moment that these muscle fibers are damaged. And what occurs then, and you have to go to the left then, that these cells can divide, and then they can differentiate into myocytes and fuse with the damaged muscle fibers. I go into detail in the next slide. But also a part of these satellite cells go back to quiescence and are again resting in this muscle fiber for a next series of muscle damage. And here you see the process in a little bit more detail. When a muscle is damaged, and this is the central area over here, then what happens is that the cell has a kind of automatic process. There's what the cell does is that it lines up the area which is still healthy. So there you have a closure of the sarcolemma, and in this area where the muscle is damaged, the central area, you get macrophages and inflammation. It's extremely important. When that doesn't happen, your muscle is not going to repair, because the macrophages, you have to remove all the damaged muscles, and then they activate the satellite cells, and these cells start to divide and start to form new muscle fibers, as you can see over here in the second panel. Oh, that's and then at the end, I don't see the marker. Oh, it's over there. Here you see the new muscle fibers formed, and at the end you have a completely, or the myotubes formed, and at the end you have a healthy myofiber again. What's important in this is two remarks. Is that what has been found with research in muscular or Duchenne muscular dystrophy, that probably the dividing capacities of these satellite cells is limited, and that probably they can not divide more than 60 times. So that at the end, this pool is not available anymore. And what's also important, when you get too much inflammation, there will be fibrosis. So, to make a long story short, and this is what I wanted to tell, is at least we can say that in Pompe's disease something happens. And we have heard that there is a lot of information and a lot of things going on with respect to autophagy. But what you can see in the upper panel, a patient who is weak and started before start of therapy, and at the end, a patient who received therapy. And here, one of our infantile patients who started therapy at 3 months, at 2 years, and here at 12.5 years of age. But what you can also see, that there is residual muscle weakness, and that there are still challenges ahead. And I also show a video of one of our adults. On the left side, you see the panel before start of therapy. On the right, after start. With this complicated story, I would at least end my part over muscle regeneration and repair, and I have 5 more additional slides, but I hope that I introduce you a little bit in what happens with our muscle building up and what is the potential. But it's a complicated story, as I mentioned. If you look at the therapeutic options, there's enzyme therapy and other options, and I would like to go very short to cell-based therapy. And I want to show you some slides that I received from Gerben Schaaf from our Stem Cell Institute in Rotterdam. And what this addresses is the capacity of stem cells to replenish. And what is known that if you isolate satellite cells from muscles in mice, and you bring these isolated muscle cells back in damaged muscle of a mice, then what you see is here on the right side that these satellite cells can divide and can make new muscle cells. If you culture these muscle cells first and you let them divide in culture and you bring them back into the mice, it's not possible. They do not divide. And as you can see on the right side, the red part of the slide, there's no green cells. So they do not divide and they do not differentiate further. Cell-based therapy is now based to see can we enrich stem cells in culture. So when you isolate stem cells or cells, satellite cells from the cells, there is a certain population in culture that retains the capacities of the stem cells. And what would be nice if you could isolate those specific stem cells and enrich this pool and use them in future for gene correction. For example, if you have POMPE cells and you repair the alpha-glucosidase gene and you could bring in back these cells, then it could be a therapy for Pompe's disease. Is that clear? So, and what they found is that if you isolate these stem cells, there is a certain subpopulation of cells that contains or that lacks a certain surface-specific marker, and if you culture those cells, then these cells without this marker are able to regenerate in cell culture. What you see here are the upper panel are cells with the cell surface marker, and here at the lower bottom you have cells without the surface marker. These cells do not differentiate in culture, while these do. So what they did next is they looked at this cell population, and what they tried to do is culture single cells of this isolated population and see what the characteristics were. And what they then found is that there is a subcell population with the marker that's not able to differentiate. There is a second population who has not the marker but is only partially able to differentiate. And the third population, and that's the stem cell population, that retain the capacity to differentiate. And then the question is, do these in vitro stem cells also have functional activity when you engraft them in vivo? So what they then did is that they injected the stem cell in a damaged muscle in mice. And what they found, and that's very good, is that the stem cells, if you bring them in these damaged mice, that they can divide and that they can repopulate the cell, and that at the start you see a few cells, they are labeled with GFP, it's a green marker to see what are the cells that I have injected. And if you wait a few weeks, you see that this population expands, and that 60% of the muscle contains these green cells with this GFP marker, indicating that these are coming from the stem cells. If you inject a second population, the myoblast A, with the marker, you don't see any green material. And if you inject myoblast B, it's 10% of the population. And what can you do with this information? What you could do is that if you add for example, the alpha-glucosidase, the collected alpha-glucosidase gene to these type of cells, you could inject it in a certain muscle fiber. The disadvantage is, of course, that you have to inject it in many places because you have so many muscles, but maybe it's something for the future. And I would like to end here.
So what can you tell us about muscle regeneration in Pompe disease?
Now, I think this is a very intriguing question. I think at present it's still open. I think not much research has been done on muscle regeneration and repair. And I think it's a very interesting topic for the future. That something happens there is clear, because otherwise we wouldn't have these children who improve. And I think this is a topic for further research. But these are the types of processes that occur that I could show.
Yeah. So we have first a question from the back, from Maurice.
Yeah, I have a question for Priya, Dr. Krishnani. What are your experiences in really late onset people with high antibody titers? Because I notice around the world that there are quite a few, and even people quitting therapy because they are over 100,000 antibody titers and not knowing what to do and really being desperate.
I think I'm supposed to stand right.
Yes, please, for the camera. Yeah, thank you.
Oh,
you should have come. Okay, sorry.
So Maurice, this is a very good question that you've asked, and I have to admit that our work has been limited more to the infants and now actually even to some of the younger children. The reason being is that the ability to monitor a response is easier or less complicated than it is in an adult. And these agents are not completely benign. I mean, they do have a side effect profile. And so what my goal is that once we have an understanding and a clear, understanding from comprehensive learning from groups of people that indeed this does make a difference, then I think we need to do this in the adults. There's no question in my mind. I think there are two parts to this. We're also seeing that some of the patients who develop the infusion-associated reactions are also the ones with the highest antibody titers. The second part, which I do believe, is that if we are able to reduce these antibody titers, maybe the amount of enzyme that's needed is not as much, or there will be more efficient delivery of the enzyme to the skeletal muscle. So to answer your question, we've not yet done the adults, but as you are aware, we have started the work in some children now, but it's to be done with a lot of caution. Thank you, Maurice.
Thank you, Priya. Dr. Plots had a question.
Priya, that was a beautiful presentation, I have to say, and the clarity of the separation of the groups is really wonderful and very enlightening. I'm curious to know whether the bortezomib-treated patients are able to sustain their protective antibodies. These are infants, for example, probably who've been already immunized with things like tetanus and diphtheria. Do you know what happens to those titers, and what do you do about it?
Sure. So thank you, first of all, for the comment. It means a lot coming from you. So it is true that when you are giving these agents, you are wiping out the immune system. So at least from the experience from the rituximab and methotrexate treated patients, which is essentially the same, it's at the level of the B cells, we are not completely wiping out the immune system because we have been checking tetanus and diphtheria toxoid levels. So we have, in one situation, there was a blunted response or a drop in titer of diphtheria titers, but it has not been across the board. And the good news is that we've been able to successfully tolerize them, which is, I think, what's critical. But are they at a period of risk? Yes, and that's why we introduced the IVIG. to these individuals because I think at some level they are immune suppressed. In the situation of the use of bortezomib, we are starting to space the duration. So bortezomib does not have to be used long term. It's really to bring the titers down. Once you've brought those titers down, I think you can go back in with the regimen that's used with rituximab and methotrexate, and then you can space it out. So we're trying to space it out with the rituximab and the methotrexate both. So to answer your question, this is not, we don't have a clear time when to say this. It's more with monitoring and with individual clinical judgment. And clearly, I think safety of the child first as we've done this. But we've been successful to date. Thank you.
Dr. Chen, next question, please.
I have a question for Nina Robin. Actually, I have 3 questions, but
Start with number 3.
it's all very short. The first question is, what are the storage material inside those autoparkinson vacuoles?
Sorry, what is the...
What type of materials inside the autophagocytes?
Yes, it's a very good question, and the simple answer is that I don't know. But it looks to me that the composition of the knockout in human and in mice is very different. So those balloon-like structure, which I very often see in humans, I never see in adults, in mice, sorry. And so one of the suggestions, again, this is very preliminary, this could be lipids. And it's an interesting new aspect to the whole story because autophagy is known to be involved in lipid metabolism. And it may well be that not in mice, but in human, this is it could be a significant problem with lipid metabolism. But I don't have a real answer.
Actually, in our early paper in Quail, we also saw the vacuoles, and it's lipid. The second question about how about the other lysosomal storage disease? Do they have the same autophagy?
Yeah, it became a very big field. It's the defects in autophagy is found now in Fabry disease, and it's looked very carefully at multiple sulfidase deficiency in Gaucher disease. Yes, so it became a very big field in the lysosomal storage diseases. And in each tissue, there is a tissue specificity of this. So in each disease and in each tissue, the way this autophagic pathology would look is quite different.
Well, last question. I think this is really important field. And is any way we can detect autophagocyte markers in the serum?
Not yet.
So we still have to do the muscle
only
biopsy?
muscle biopsy. Unfortunately, it's only muscle biopsy.
Thank you.
Thanks. Next question, Dr. Kubel.
Yes, that.
Mrs. Han, I have a question for Dr. Rabin as well. And I've struggled over the last 5 years interpreting the very elegant data from your laboratory.
Me too.
Okay, we're in this together. But so for me, from the point of trying to develop new therapy, I've been wondering, and I think you're closer to an answer, is it more important to suppress the abnormal autophagy, or is it sufficient to really replace GAA more effectively?
I think once the autophagic buildup occurs, there is very little can be done in terms of with therapy, enzyme replacement therapy. I don't think it really resolves autophagic buildup, so the best way is try to prevent it. Meaning start therapy extremely early, that's why I'm so much for newborn screening.
But there is hope with very early and effective therapy, there's hope at arresting and preventing the autophagic buildup.
Yes.
There is an abnormal gene, an enzyme, a GAA that's deficient in function, and we have understanding that there's abnormal autophagy. Do you have any sense of the relation between the two? So, enzyme replacement therapy is directed at the abnormal gene product. late in the disease, we know enzyme replacement therapy will not affect autophagy. But enzyme replacement early in the disease process may have an improved clinical outcome. Is there potentially an effect of enzyme replacement therapy on autophagy? What's the relationship between these three corners?
Again, I don't have a good answer for that, but my understanding of the process is that if you start therapy when early enough, when the lysosomal pathology is resolved, when essentially you maintain the normal level of glycogen accumulation, which is no accumulation at all, then the autophagic buildup would not develop.
So there may be something in terms of the accumulation within the lysosome that may be a trigger for abnormal autophagy.
It's possible. It's possible that the position of the lysosomes within the muscle fibers, and possibly even a small breakage of the lysosomes, send a signal to autophagosomes to get rid of this organelle, because it is a damaged organelle, and that is a function of the autophagic pathway, to get rid of the damaged organelle. And curiously, we actually very often see the lysosome inside autophagosome rather than the other way around, suggesting that it may well be that some signal is sent by the lysosome for the autophagosome to come in and attempt to engulf the damaged organelle.
Yeah. So the point is critical in that if it's a single defect where enzyme replacement can be applied very early, and the lysosomal defect or problem addressed very early, then autophagy can be addressed early. If there are multiple defects, then we have a much more complex problem.
Exactly. That's correct.
We can take one more question and then we go for the coffee break. And if there are more questions, you can return or come directly to the speaker, because otherwise we're running late on the roundtable patients, and we don't want to do that. So please.
Okay, one more question for Nina. So my understanding is that the autophagic buildup occurs secondary to glycogen accumulation, and if the degree of glycogen accumulation is determined by the amount of residual enzyme activity, why do you see so much autophagic buildup in the late onset patients? Even at infancy, you seem to see a lot of autophagic buildup. How do you make sense of that? It's very...
Okay, this is how I see the infants, and it was, in Anne's presentation, you could see that muscle fibers begin to develop in utero. And so the infants are born already, and some of them who are CRIM-negative and who do not have any enzyme activity, they are already born with severely damaged muscle. And the damage is mostly lysosomal, which means that this lysosomal pathology and lysosomal glycogen accumulation occurred well before they were born. Now, why at some point during embryonic development autophagy didn't kick in? And the answer for that is that there is no need for autophagy in embryo because they have a constant food supply. So the autophagic system, the process, works, if any, at a very, very minimum level. And it was actually shown in experiments in mice that the autophagy is at extremely low level during embryonic development. So you end up with infants being born with already severely damaged muscle fibers because of the lysosomal pathology, but the autophagic buildup is not there.
Maybe I misinterpreted. So it looked like you had some late onset patients that were identified by newborn screening, and you were showing microscopy at a very young age, a few months of age, and you still see a lot of autophagy. I just don't know.
Not a lot, but yes, there is some signs of autophagic accumulation in late onset disease. Yes.
Just make very clear, because now we are talking about late onset. We are not talking about, I don't know what we're talking about, because late onset does not have any definition. Some people say late onset starts at 0. Other people say late onset starts at 25. So your question cannot be answered unless we know the age of this patient.
I guess by late onset I mean they have some degree of residual enzyme activity.
A degree of a disorder.
Residual enzyme activity.
There is no definition yet, I think, for late onset. I don't want to go into discussion because we go for coffee break. Thanks to all the people. And can you please take your has been playing, but I think later also you can probably still maybe have an occasion to look at it or whatever. We will now start with a session about gene therapy, and each of the speakers has only 15 minutes, which is extremely short for such a very complicated subject. But we still wanted to talk about gene therapy because that's the maybe a therapy for the future. To keep it short, I start at the end, and I give a very brief introduction just to kind of summarize where we are and what we are doing. This is again to emphasize how important patients have been talking to the doctors and talking to the scientists about their disease and what they should do. Tiff, you recognize still yourself? This is Tiffany House. Oh, another. Oh, that's also Tiffany House. All right, we have 4 speakers. Alphabetical order. You can read it. I'm not going to read it to save time. Yesterday, I said we have DNA. That's the information. And then at the end of the process, we have the GAA. That is the protein. First, we have been talking about enzyme replacement therapy. This is again the same cell I showed yesterday. The nucleus contains the DNA and the information, and the protein does its work, which is to degrade glycogen into glucose. Okay, first enzyme therapy. Enzyme, I have to push it. Enzyme therapy. This is enzyme therapy. You add an enzyme to the outside of the cell by intravenous infusion, and there it goes in, plop, plop, and it ends in the lysosome, and you have your activity. Now we go to gene therapy. That's completely different. Gene therapy, you're not adding any enzyme, you're adding information. How do you do it? You use a virus. So the gene is now inside this viral particle, and then the particle infects the cells, and we have more particles to infect, and then the particles containing the information that's missing or wrong is now going to be delivered by the virus to your nucleus, to the DNA, and then, of course, when you have the correct information, you can start production of acid alpha-glucosidase, and you correct the defect. So that's the difference, and how to do it. A single cell is easy to do. I'll show. It always takes a little bit to load this one. Okay, if you start with the single fertilized oocyte, it's easy, because you inject in the oocytes, and as I've shown yesterday in the movie, automatically at the end you have an embryo or a human being. This is how it went with the product we made from the rabbit milk. The rabbit oocyte was injected, and then the rabbit oocyte produces the alpha-glucosidase, and the alpha-glucosidase went in via infusion. The different speakers, they use different factors, vehicles to deliver the genetic information, and Dr. Amalfitano is using the adenovirus, but maybe also he'll be talking about adeno-associated, I don't know, we will hear. Dr. Burns is using the adeno-associated virus, Kuber also adeno-associated, and then we have a presentation using another virus, the lentivirus. This is it. And the floor is now to the first speaker, Andy Amalfitano, Dr. Andy Amalfitano. Sorry about this. I know you too well, you know.
Okay, well,
15 minutes.
15, okay. Get my clock going. Give me a hand signal if I go too long. So good morning, everybody. Thank you for staying and waiting, and I appreciate the invitation to come and talk. Briefly, I'm at Michigan State University. Here's an aerial view of the research complex where I hang out. My laboratory is here. This is where I trained as a medical student, and I was at Duke for a while, and I returned back a few years back. I have a laboratory in this building here, and I insisted I have a view of not only the football stadium, but also, since I was at Duke so long, I had a view of the basketball stadium, so I get to see that from my office now on top of the atomic collider and everything that's out there. Michigan State is a top research university in the world. We have a number of things going on there. I teach at the medical school here, in which I teach actually 600 doctors in one class. We have 2 medical schools on campus, and I guarantee you that all of them graduate knowing exactly what Pompe disease is, and that they are not going to miss it if anybody happens along. So, yeah. And I'll tell you, the lessons of Pompe disease, all the various patient stories that you hear about, how I saw 8 doctors and everybody missed it, and then we finally got to the right doctor. The story goes on and on. I use that, actually, the medical schools asked me to come and talk to the students on their first day of school to tell them that this is what their job is to pay attention to what the patient is saying, otherwise you're going to miss it. And so we use that as the premier example. So your stories are training the next generation. I see patients. I continue to see patients. I treat patients with Myozyme in my clinic. There's a hospital here where I see patients, and some compelling stories are online. If you YouTube me, you'll see some patient stories there that show that once on enzyme, some individuals show some dramatic recoveries and reversals, in fact. So there's a lot of promise there. Now, I'm going to focus on my studies today in regards to gene therapy, and as Dr. Reusser told you, what we're trying to do here in this angle is not to replace the enzyme, but replace information. So in your cells in your body, if you have Pompe disease, at some level you are missing genetic information, and what I'm going to try and just show you here is how we can deliver genetic information, not correct the existing issue. That's a different and more difficult target. And if you know me, I'm trying to get to things that I see as a clinician the most likelihood of getting to a patient as rapidly as possible. So I've been gearing my research to that goal always, okay? And some things are more difficult than others. Adding the gene information rather than trying to correct existing is easier. And so that's what we're trying to do, and hopefully we can add enough of that genetic information that it produces the protein that's missing in the body. So as you heard, we're using a virus. This is the virus. It's an adenovirus. This is a virus that will give you a cold if somebody sneezes on you. But we've genetically engineered it in the lab and taken out portions of it that will give you a cold and rather turn it into a vehicle to deliver genes at our will. And in fact, we can manufacture this at high level. In fact, thousands of humans have received these type of adenovirus vectors by all kinds of routes. I have been to factories that can manufacture this, so it's not that this is something we just do in the laboratory. I'm convinced that if we come up with the appropriate target and the appropriate patient population, we can move this rapidly into the clinic. In fact, some of the viruses that I've developed are in human trials right now for other diseases like cancer. And we've moved into a variety of those sort of avenues, so I'm pretty convinced of that. So here is an example of what you can do with these viruses. This is a virus, one of the viruses that contains a gene that simply turns the cells blue if it successfully delivers the gene. And if you intramuscularly inject this virus just directly into your muscles and look at the muscles, you can see blue cells there. And you can see that in some cases, this is 84 days after injection, the cells remain blue for a long period of time. But I show this also to show you that even with a high dose of the virus, not all the muscle cells are being taken up. It's only within the vicinity of the injection site, and that's a problem, especially when you consider that Pompe disease affects all the muscles in your body. Okay? So a simple intramuscular injection is going to be limited, and we've shown this in a number of studies that certainly we can deliver the GAA gene into muscle cells, and we can get those muscle cells to correct locally, and they even can produce a little bit of enzyme and secrete it, but it's not enough to correct the entire body of all the muscles all at once. And I cite papers here, you can go look them up, and that's where I draw my data. So widespread correction of the glycogen is not possible by limited direct injections. And as we heard from Dr. van der Ploeg, 40% of our mass is muscle. Those are a lot of cells that need to be corrected. That's why the dose of enzyme is high. You have to deliver it to a lot of muscles at once. It's one of the more daunting tasks, actually, in modern medicine. So how could we do this? Well, simplistic way would, why not just inject the virus into the bloodstream and let it go to all the muscles? Well, our bodies are designed that does not happen. If that were happening, we'd be infected with all kinds of organisms. Our blood systems and our circulatory systems are closed systems. Nothing leaks out of those. Small molecule, you heard about how enzyme replacement therapy, even when you get that infused, that small little molecule has to get out of the blood vessel, traverse several cell layers to get to the muscle cell. And that's a small little molecule. A large thing like a virus is blocked. And so we can't just simply deliver the virus throughout all the muscles in the body. But what nature allows us and what anatomy allows us to do is that our livers are actually natural filters and allow bigger molecules to get there, and that's their job, actually, is to act as a filter. And what we notice is that when we take this virus and inject it into the system, the circulatory system, it actually goes to the liver. And here's a liver before injection of the virus. Here's the liver after we've injected the virus, and all those blue cells are where the virus successfully went in and it effectively delivered genetic information. And you can do this with these viruses. You can change the liver from light blue to black so we can control the dose. And you can do this not only in mice, you can do this in other animal models. We've done it in quails, we've done it in dogs, we've done it in non-human primates, we've done it in baboons, and in fact I'll show you data in humans that's confirmed that you can deliver genes into the human liver as well with this virus. So many years ago I had the idea, back when I was at Duke, could we deliver the GAA gene to enough liver cells that they would actually produce enough enzyme to supply the body, all the muscle cells in the body. So we turn your liver into an enzyme factory. So rather than Genzyme producing the enzyme or one of these other companies, we let your own liver make enough enzyme for your body. That's the goal of our sort of flavor of gene therapy. Okay? The goal is, can we produce enough enzyme to get all the muscle cells to take up that enzyme and clear out the glycogen? And one thing about this that's interesting is that your own liver produces it, which means your own liver would glycosylate it to its sort of specifications, which probably is better at some level, although I don't have exact proof of this, than maybe a Chinese hamster ovary cell producing the enzyme and then having it infused in your body. So there's other benefits here that come into play. So, can this be done? In fact, this is one of the first and few experiments I've ever done that worked the first time. We injected this virus containing GAA into the GAA knockout mouse that were kindly provided by Dr. Plotz and Dr. Rabin from NIH, and let me tell you, that is not a small thing. I've run in plenty of other investigators that don't share resources like these animals that are very, they took a lot of effort to develop that, and they were very willing and provided these animals for us to do our studies. Here's what some of the data, it's old data. It's actually, in some ways, this is very distressing to me that it's old data because we have information here that's very old and yet we're not in clinics with it, and I'll explain why. But in 1999, we showed that if you take animals that are untreated and you give them this virus by a simple intravenous injection and then wait about 12 days later, the glycogen can be reduced significantly in all the muscles in these animals because their liver is producing the enzyme. It's almost like you have a bag of enzyme hung on your body, but you're walking around with it 24 hours a day, 7 days a week. The liver is constantly producing the enzyme. So you can imagine that can affect a pretty tremendous change. And here you can see the before and after. The pink is the glycogen significantly reduced in all these tissues, although there is variation from fiber to fiber that Dr. Rabin noted, and we saw it as well. Here's the heart muscle. Here's a normal heart muscle from a mouse, and here's one of these untreated animals, and within 4 days you could see clearance, starting to see clearance, but by 12 days, complete glycogen reduction in the heart, which turns out to be the most responsive. One thing in science, you want to have things replicated by other groups, and Dr. Byrne, who's here, also showed this, and we've also shown this ability of using the liver to deliver enzyme using other viruses, including adeno-associated virus. So the paradigm is there. We've gone on and shown that we can actually sustain this using various flavors of this virus for long periods of time. This is almost a year out, and we could determine that the liver could continue to produce the enzyme at high levels. We also showed, if anything, these gene therapy models for me allow me, when I see patients, to learn things. And one thing I learned here is that, again, these are mice provided by Dr. Rabin, Dr. Plotz. We have actually CRIM-negative and CRIM-positive mice, and we knew very early on that CRIM-negative was not good. And in fact, in gene therapy, the same thing comes into play. If the animal responds to the gene transfer and the protein is foreign, you don't get as a good response. The animals don't produce as much enzyme, and in fact, it's much less than if you're CRIM positive, in which we see our most promising results. We showed that we could keep the glycogen down after a single treatment with this virus. So you imagine one injection, the virus goes in, and within 30 minutes it goes into the liver, and then that's it. But that gene information in there is constantly churning out, producing enzyme, and that maintained those glycogen levels down for as long as we did the experiment in the CRIM-negative or CRIM-positive type animals. And in fact, here's that terrible rotorod you heard about earlier, where we put these, we put the animals, we put the animals on, and they sort of hold on, and as this thing speeds up slowly, they eventually fall off and hit the little trigger there, and we time them. And we proved that with this, 5 minutes left? 4 minutes left, okay. We could determine that their strength was increased as the glycogen went down. We showed that we could, in fact, if you looked at one of these studies, animals that were weak, that were treated with this, actually gained strength, so we could reverse the process in these animals. And actually in older animals, although they're not, mice and humans, there's limitations there, but certainly in older mice we could reverse the glycogen as well. Here's an example of how I see this translating. If you take, this is an animal that's untreated, if we give a low dose of the enzyme, too low to correct the glycogen in their heart, you see this is the glycogen levels elevated, we give them a low dose of the enzyme, really don't see much glycogen reduction, maybe a little. If we give a low dose of the virus, we get a little reduction of the glycogen, but if we combine the two, low dose of enzyme and a low dose of the virus, look what we can do here. And that's how I envision this maybe moving into the clinic, that it can be used as an adjuvant to produce enzyme at a level that will make maybe the need to have less enzyme therapy because your body will be producing some of its own as well. That's unpublished data. Here's the problem. As we did our studies, we started doing dose escalation studies. And you can see here that as the dose of the virus increased, we could start seeing at higher doses the heart glycogen decreasing, so that's good. Took higher doses of the virus to get the skeletal muscle to respond, okay? So more enzyme was required to get the skeletal muscle to respond. And these doses are predictive to what humans would be. Now, I can tell you that this is what it translates into particles per kilogram. In a non-human primate, up here is where you get into some serious toxicity with these viruses because your body doesn't know that they're engineered viruses. And in fact, in human trials, several human trials have been studied in which intravenous injection of adenovirus vectors encoding other genes have been performed and published at this dose and have been done safely. In fact, up to 50 patients have been delivered this way without any problem. Unfortunately, at the University of Pennsylvania, one patient received a dose right there, and that child ended up dying. There's a lot of reasons for that. One of the reasons was that the trial was not conducted properly, and the PI and the whole group got into big trouble. They violated their IRB protocol. They delivered that virus to this boy who actually had an underlying liver disease that wasn't controlled, and that was why they violated their protocol. They shouldn't have gave the virus. And if you don't believe me, you can read the article from the PI. He actually had to publish an article describing the mistakes they made in that trial. I bring that up for two reasons. First, it's totally stalled our ability to move forward clinically. Secondly, I think as you hear from Dr. van der Ploeg and Dr. Kishnani about the work they've done in humans, their efforts to do those trials perfectly are very important because any problem could have set the whole field back. And so their efforts may appear sometimes not fast enough, but they're doing that for a reason. Now, the problem that we're encountering is at these higher doses we could run into toxicity. You inject the virus, you've got a number of systems in your body that want to respond to the virus. There's Kupffer cells, macrophages, antibodies. There's proteins in your blood called the complement system. In fact, the Nobel Prize was just given out last week to people who study these systems. And so we have investigated the last 5 years, and these are all the papers that I can cite that we've published, trying to understand what these toxicities are and how can we mitigate them to make this safer. Okay? And it's a complicated process, and that's what those papers basically add up to. One way we can do that, and we've published this, is that we could simply, before we give the virus, I can imagine giving a high dose of an immune suppressive called dexamethasone. This is a steroid that suppresses these innate immune responses. And in fact, here's an example where IL-6, this is a compound that, if you ever get a flu, you feel kind of icky, yucky, this compound is doing a lot of that. If you don't give this drug, this virus, when you inject it, gives you high responses of this IL-6. If we give this drug preemptively, we can mitigate that problem. And so here is a simple solution. This Decadron is available, and we can foresee using this in future clinical trials. Here's the same data again. I'll just go through that. Here's another thing we've published in which we can actually modify the virus and stick onto it, actually engineer molecules that defeat the innate immune system and mitigate its ability to be triggered, and we've produced that as well. Finally, we're in collaboration with Phil Eng and Baylor, and what he's done is found a way that actually, in a living individual, we can deliver catheters to block the liver and just infuse the virus simply into that organ without it going systemic through the body. We can use lower doses and still effectively turn that liver blue more effectively. All these things are going to be brought to bear in our studies. And so what we've started to do now with funding from the AMDA and patients, we are going to do a non-human primate study. This will be in baboons, in which we've engineered a virus that we actually made a baboon version of the GAA gene. We're going to package this up into this virus. We're going to use this balloon occlusion catheter method that Dr. Phil Eng has developed, and we're going to test and see how can we deliver the gene into a large animal like that, and can we improve the safety of it using the strategies I've just outlined for you. And we will proceed in that direction. And in summary, this is Dr. Pompe. I hope he's smiling on us. I've read more about him, and the more I read, the more I just, a phenomenal person. Gene transfer is feasible. In Pompe disease, it can reverse the problems. Various viruses that you're going to hear about have limitations. Adenovirus has many, but there are ways to proceed and mitigate those problems, and I think we can move forward with this in some fashion in the future. And I'll acknowledge everybody, obviously, my lab, people when I was at Duke and at Michigan State, Dr. Chen's lab, Dr. Kobril lab works with AAV, Dr. NIH, the corporate support from Genzyme, private support from AMDA, and patients. Thank you.
Fantastic.
There we go. All right, thank you very much. I want to thank the AMDA and the IPA for this invitation to talk about the human clinical trial for Pompe disease that we began at the University of Florida recently. And I'm going to try to focus almost exclusively on the clinical data from the first 3 patients enrolled in this study. But I think it's important to give a little bit of perspective. So by means of a few slides about background. So my clinical background is as a cardiologist, so it may not surprise you that our initial interest is in the heart disease in Pompe, and we will continue to pursue the gene therapy strategy that will impact on the heart. But in a way, we wanted to pursue a stepwise approach in which we can get some clear-cut answers when we're in a period where patients are having important clinical benefit from enzyme replacement therapy. Just to show you the duration of time it takes to kind of move a project like this along, we first began this project actually in the mid-90s, studying this virus, adeno-associated virus, as a means to permanently transduce skeletal muscle cells. And it's been a journey to get from the early period of the work with AAV and really the significant contribution of the group at the NIH to develop an animal model that enables preclinical testing and paves a pathway to human clinical studies. But in general, this 10-year timeline is unfortunately common in the field of biologics development. It really was, in fact, quite remarkable that Myozyme was licensed in the short period of time it was from the early development through the commercialization. And the Myozyme studies were so encouraging because there was a high rate of survival in the patients who began therapy under 6 months of age compared to the natural history cohort that Priya described as an important comparator population. But the primary outcome measure of this study was not survival alone, because none of the patients began on assisted ventilation. And the primary outcome variable was, in fact, ventilator-free survival. I show you here actually at the end of the study any ventilatory support, because many of those patients, although they had some non-invasive ventilatory support, went on to receive assisted ventilation. And so that got us thinking about why was there this decay in freedom from independent ventilation. So when we look more long-term, including the patients who were in the 1702 study, some of whom were on assisted ventilation, and those that were not, separated here into two groups, you can see that a proportion ultimately either had loss of independent ventilation or died, and those in the yellow box were those that are alive and free of assisted ventilation. And so really it was an unacceptable proportion of those that lost independent ventilation. We wanted to understand that more. So Mary Rucker in the lab back in 2004 confirmed what had been really part of the initial description of the mouse model, but we looked in this circumstance at early correction of the accumulation of glycogen in the diaphragm. So here PAS staining shows glycogen accumulation, and that we quantified that over in this panel, and you can see that as the age increases, this worsens. But the muscle is not the only part of the story. The muscle is controlled by spinal motor neurons that exit the spinal cord. Those that control the output are shown here in red. And then the motor neurons actually receive feedback from the targets. And these afferent fibers are important for controlling the magnitude of the contraction that is initiated in the lower motor neurons. And when we looked in the mouse model at the region that controls respiration, because of this finding in the clinical trial, we saw that as the mice age, there's an increasing amount of glycogen in the cervical spinal cord. So the GAA animals shown here in red and the controls in blue, and that significant amount of glycogen had an impact on ventilation. And we tested that in the mouse by doing a mouse pulmonary function test. And the way that's done is to ask the mice, kindly, to breathe faster by giving them a little bit of CO2. So as you probably all know, as the CO2 builds up, there's a signal to the brain to improve ventilation. And we saw that in the control animals, they responded quite nicely to CO2 and reached a maximal minute ventilation. And in the Pompe animals, this was severely reduced. And there are probably many reasons for that. The control of breathing is a complex process that involves some control areas in the brainstem and also the spinal cord. So then why gene therapy in Pompe disease? And the vehicle that we chose, as you heard from Andy and Dr. Reusser, there are a number of vehicles to traffic this gene into the cell and hopefully get it to stay there and do its job long term. There are a number of examples now in neuromuscular disease and in the central nervous system, including the eye, where AAV vectors have been used clinically with great success. And in fact, last week we had a meeting of over 500 investigators, and speakers from many countries where, particularly in the EU, there have been very notable successes in gene therapy, and to try to come to a consensus as to how to continue this concept that molecular medicines are probably the most specific medicines we can use for inherited disease. And so it was really actually a very successful conference with a renewed hope that these strategies will become clinically useful. But in Pompe in particular, as has been said throughout the meeting, we know there's a single gene causing a single disease, and we are beginning to unravel, although we learn there's more complexity than we probably anticipated in the beginning, what the pathophysiology really is. But AAV vectors delivered to muscle cells, at least, seem to last the life of that muscle cell, so that's encouraging. We can use a single-dose therapeutic that may have a systemic effect if we reach the point of systemic dosing. There are a number of issues, as were discussed earlier, about antibodies, and those antibodies interact with the protein in the circulation, but they don't appear to interact with the protein when it's protected inside the cell. So GAA made within the cell can make its path, as it normally does, into the lysosome, provided that the lysosomal mechanisms for trafficking are not completely disrupted by prior muscle damage, and certainly the ability to, for various muscle fiber types to do this seems to be similar, unlike the ability of some fiber types to traffic. And then lastly, we want to reach these motor neurons because ERT does not impact on the central nervous system aspect of the disease. So it's a simple design of the vector. This vector, called adeno-associated virus, is devoid of all of the viral genes that are required for it to live independently. So it requires a special manufacturing method. It puts this DNA inside of a little coat, which is shown here, and this is what the package looks like that's delivered into the muscle or into the bloodstream. And in the example I'm going to show you in the first three human subjects, the dose has been delivered into the muscle cells with the goal of having some of the virus make its way up the nerve into the spinal cord to reduce glycogen and improve function in the phrenic nerve. And here's a recording from a mouse phrenic nerve in the untreated and treated animals. And you can see in an individual EMG that the ones dosed with AAV have restored the amplitude of the phrenic output. We delivered those via the diaphragm, so there was an impact on respiration, which I won't go through, except in this example where we more specifically targeted only the spinal cord. So in this example, we dosed the cervical cord and examined both in the region of the dosing and distal to that, and you can see that the activity with the vector that carries GAA is present in the area that was dosed and not, but only a little bit influenced down below. The control virus didn't impact the activity at all, and here are the control animals, which shows that one can completely correct the level directly in that space. And then what happens to ventilation when you do that? We saw that the ventilation, this red dot, is actually the unaffected animals. So only with motor neuron correction, still with a pathology in the muscle itself, can you restore ventilation. So the muscle can work if it receives the appropriate signal. And the other deficit we saw in these animals is that the rate is abnormal. As the animals age, their ventilation is influenced by their breathing rate, which declines, and it's restored after treatment. So we now had to test this concept in both mice, rabbits, and in a non-human primate to achieve permission to proceed with humans. And these are some experiments done with the group at UC Davis, where we looked at longevity of expression in newborn or preterm macaques. And now these animals are actually older than 36 months, so we think this is essentially permanent. And this gave rise to the clinical trial, which I'll describe. It's a phase 1/2 trial, which means it's a safety study. We tried to choose a dose level which would be informative about the potential efficacy. Catherine Ma, shown here, really did most all of the preclinical work, and Salim Islam, shown in the lower right, is the surgeon, the pediatric surgeon, who gives the dose into the diaphragm with his expertise in this approach, and I'll actually show you how this is done. And really, I think I've described essentially the rationale for the study, but I wanted to point out that part of this is that we are using a concomitant therapy. All the patients remain on enzyme replacement therapy. We know that one of the problems is loss of independent ventilation, and that to enter into this study, patients were required to have failed independent ventilation and have a tracheostomy and full-time mechanical ventilation. So in some ways, we had the most challenging patient population who already have significant accumulated disease, but no significant impact of ERT in the central nervous system. And then we knew from other studies that we had been involved in that this was relatively safe, and in fact now over 100 patients dosed with AAV in studies like this. So we designed a 2-cohort study at 2 dose levels, 5-fold different in the higher dose cohort, based on the safety data that was accumulated in the preclinical experiments. The vector we chose also had the greatest number of patients previously studied in those other studies I mentioned, although I think there are always better vectors that we determine in the laboratory can be brought to the clinic, and there are second-generation materials that we think will improve this, and I hope Dwight will talk a little bit about that in his talk. But the study design is shown here briefly. It's essentially a 1-year safety study with some important efficacy outcomes, which I'll show you. And maybe just to emphasize that the patients do continue enzyme replacement therapy, and we also had a pre-treatment period of inspiratory muscle strength training to try to gauge the effect of rehabilitation prior to dosing, rather than have it happen independently after dosing, so that we could see whether this effect that we're measuring could be improved just by the rehabilitative therapies alone. And as I said, the outcomes are safety. They're shown here. I'll go quickly through that so that you can see what the dosing is. There are secondary outcomes related to function, and those are the data I'm going to focus on. This will give you an idea of what we're after. So this is the 3D view of the diaphragm, and you can see that in a normal pump function, it has a very substantial excursion. And this is looking from the side, so this would be kind of the surgeon's view looking through the ribs at the diaphragm muscle. It's possible, although it's quite thin, to do a direct intramuscular injection like all of the other muscular dystrophy trials where AAV has been delivered to muscle that have been done thus far. And now here is the appearance to the surgeon. This is looking at the surface of the diaphragm from the chest. And if you look up here at the top, you'll see a needle come in from the chest wall. That's the needle that has the gene therapy vector in it. There's one dose that's already been delivered over here. The needle enters the thin diaphragm, and this diaphragm is not at all normal. It looks very pale. Blood flow is diminished when on assisted ventilation because the diaphragm doesn't need to do any work. And the vector is injected intramuscularly. You'll see here it gets a little more pale when the dose is delivered in a second. And here comes the dose, and that spreads throughout the muscle. And in studies in 3 kilo animals, we determined that these 3 injections across the surface of the diaphragm actually spread to most of the diaphragm tissue as this is allowed to spread further after the injection. And so, as I said, the key outcome is safety. So here are the laboratory data, the change from baseline in either the liver function test or the muscle CK marker, and there's essentially no change. And in fact, some patients it decreased. We also looked at antibody titers. There's really no change, at least over 90 days, to exposure to GAA from the vector. That's a very small additional amount of GAA compared to what's received. And when we look at some of the function parameters in the first patient to pass 90 days, we measure respiratory function in a number of ways on patients on the ventilator. One of them is peak inspiratory flow, so this is independent inspiration without the ventilator. No change between screening, baseline, and 14 days. And then we can see what happens at later time points by adding a load. And the load compensation shows that actually the patients actually gain some strength even at 14 days, but that's very minimal. What we really want to see is after 90 days what happens. This shows no change in the maximal inspiratory pressure, but what we really saw was a significant change was the maximum voluntary ventilation. So this is independent ventilation compared to baseline and after dosing. And you can see that the two patients that have crossed 90 days have had significant changes in maximal voluntary ventilation. And in terms of time off the ventilator, this went from a pre-dosing time period of less than 5 minutes to post-dosing of 75 minutes, and we think that that will continue to change as we modify the technique for ventilation. So I'm going to end there and thank the patients who volunteered for the study, some of whom are even Florida fans, and particularly the group. It takes a large team to put this together. This is our group at the Gene Therapy Center, and I want to reiterate again, aside from our group, a lot of this approach was learned from the enzyme replacement studies and all the investigators in the pivotal studies, as well as Nina and Paul and their continuous support from the NIH. So I'll stop there so we can go on.
Okay. So our topic has been following the lead actually from Dr. Plotz and Dr. Raven several years ago, that receptor-mediated uptake of RhGAA was impaired in Pompe disease. So this is an outline for the presentation. Again, I'm just going to review briefly some of the limitations of ERT, talk about this problem of low receptor-mediated uptake of myozyme, and then explore an approach to increasing the receptor-mediated uptake of GAA in mice with Pompe disease, and demonstrate some benefit from that both in an experiment with ERT and also with gene therapy. We'll look at this as an adjunctive therapy and then talk about future directions of what we might do in a clinical trial. So, of course, this is a biochemical defect with deficient acid-alpha-glucosidase in the lysosomes and glycogen accumulation, and this is a gross oversimplification of the pathogenicity in Pompe disease. We appreciate that. But what we've tried to address then is receptor-mediated uptake of GAA in the muscle. So during ERT, recombinant GAA must be taken up through the mannose-6-phosphate receptor into muscle cells and then trafficked to the lysosomes to be active. And we know that the response to ERT has been less than optimal in many patients. In infantile Pompe disease, there have been motor developmental delays, residual ptosis, respiratory insufficiency that indicate both muscle and central nervous system involvement. And in late onset Pompe disease, we know that some patients have developed severe pulmonary insufficiency and have residual gait abnormalities despite adherence to ERT. And you can see the studies that have been published to really document these limitations. Now, one view of the problem then, at least one part of the problem with ERT is very low mannose-6-phosphate receptor, or CIMPR, as I'll refer to it from here on, in the skeletal muscle. So in panel B, this shows a Western blot from Dr. Raven's laboratory, and you could see with the arrow that in an individual mouse, the receptor level is much lower in the muscle, or M for muscle, than in the heart, diaphragm, or liver. And then you could correlate uptake of GAA in these mice, and it's taken up very avidly in the heart, diaphragm, and liver, and very little in muscle. And the result of this has been that ERT doesn't work very well at clearing glycogen from muscle in GA knockout mice. We can see in the levels of GAA that the activity is greatest in the liver after 4 weeks of ERT, less in the heart and diaphragm, and very low in quadriceps and gastrocnemius. And these are very important muscles that are severely involved in Pompe disease. So we've developed an approach then to this problem with low receptors. And the idea was in the schematic that if the receptor was very low at the plasma membrane, then we could increase it with a drug if we found the appropriate drug. And then uptake of GAA would be increased, targeting to lysosomes would be increased, and the efficacy would be better. So we found in the literature one such drug. This is Clenbuterol. It's a beta-2 agonist that has been used widely in asthma for bronchodilation effects. And its toxicity is very low. It's tolerated well. So look at this bottom panel with the box and whisker graphs, if you will. In the lower right, we can see the level of IGFR2, which is insulin growth factor type 2 receptor, is increased in the masseter muscle, which is a type 2 myofiber muscle in rats by Clenbuterol treatment. And actually what this data reflects is that there's activation of growth hormone axis, that IGF-1, IGF-2, and their receptors are increased. But the really important thing is that the IGF-2 receptor is also CIMPR that's needed for uptake of GAA, and also that this is a type 2 myofiber that's generally resistant to ERT and Pompe disease. So given this, we formulated a hypothesis saying that increasing the receptor with a beta-2 agonist would actually enhance ERT. And I've mentioned the mechanism involved already with IGF-1 increase, muscle hypertrophy, and also an associated mannose-6-phosphate receptor increase. And we've actually generated data that show that skeletal muscles uniquely respond to this increase in receptor from specifically Clenbuterol treatment, and that this increased receptor expression increased uptake of GAA. So I'll show you that data in a minute. Here's the experiment. So 4 groups of GA knockout mice obtained from NIH, courtesy of Dr. Rabin, and they were treated with 4 weeks of ERT with the drug, with ERT alone, with the drug alone, or untreated. We monitored rotarod performance, as have been mentioned earlier today, the amount of time they can stay on this rotating rod, and the total time reflects their neuromuscular performance, if you will, and also weight to try to detect muscle hypertrophy. And after 4 weekly doses of ERT, actually accompanied by antihistamines to prevent an anaphylaxis response, we analyzed skeletal muscle, the heart, for biochemical correction reflected by GA activity and glycogen content improvements. And here's a summary of that data, where on the left in the dark bar is the glycogen in the muscle of the mice treated with the combined therapy, ERT plus Clenbuterol, and on the right, on the pale yellow bar, you can see the glycogen from ERT alone. And I want to emphasize that ERT is not very effective at standard doses in the GA knockout mice. And we could see the improvement, the difference in the 2 bars in the diaphragm, quadriceps, and tibialis anterior. The tibialis anterior is a muscle comprised primarily of type 2 myofibers that are generally resistant to ERT. So this is an important result, we think, both because several muscles were corrected more effectively, and also because specifically type 2 myofibers responded better to ERT. Then another question that's been asked very astutely by our reviewers is whether the drug alone has some effect on glycogen content in the muscle, and it really doesn't have a significant effect during this treatment at reducing glycogen content in the muscle. So we see a synergism or combined effect from both ERT and the beta-2 agonist. Now, actually, this is a gene therapy session, so let's get to that point. Asking the question whether enhanced receptor expression would improve gene therapy. And so this is a schematic of the liver as a depot, actually an approach developed by Andy Amalfitano several years back, and we were fortunate to be working next door to him. So we adapted this approach to our AAV vector, and when we advanced using AAV8, we found high efficacy from this when the vector is given at what would be considered a fairly high dose. these liver cells produce an excess of GAA, which enters the circulation at very high levels and then is taken up in muscle cells throughout the body. And we can see it achieve a significant correction of glycogen in the muscle cells after this liver-specific expression of GAA. Now, I'm specifying liver-specific expression because this vector contains a control switch that only makes the GAA at high level in livers. And we've also taken advantage of a phenomenon where liver-specific expression induces immune tolerance, and this prevents an antibody response in the GAA knockout mice that would otherwise prevent this therapy from being effective. So that's something that could be exploited clinically, perhaps in the future, inducing immune tolerance with the liver-specific expression of the enzyme. So this shows data generated by Luis Franco in the lab in 2005. It was published where you can see the incredible glycogen vacuolation in the GA knockout mouse heart, and this is, I think, convincing PS staining. There's a very high level of glycogen in the lysosomes in the untreated mouse. You can see on the left, your left, yes, the wild-type mouse heart, and then on the right, the GA knockout mouse heart after treatment with the AAV vector that produces high levels of GAA in the liver. And this is out 6 months, so this is a long-term, what might be considered a long-term treatment. We've had used an alternative approach to gene therapy which has different characteristics. So this is actually delivering an AAV8 vector at fairly high particle numbers, so a high dose. And with that high dose vector treatment, it will enter both the liver and muscle cells. Muscle cells at lower copy number, less vector in the muscle cells, but still it's there. And in this vector, the control switch or promoter is muscle specific, so it only makes GAA in the muscle. Now you could see the level of gene, what we call transduction or infection of cells in the heart, quadriceps, and gastrocnemius on the right here from the microscopy. And the very dark cells are actually good. This shows gene expression in all these cells, and again, this was a long-term experiment. Preservation isn't perfect. This is untreated Pompe disease muscle, but you can appreciate, say, in the quadriceps that many of the muscle cells have taken up the gene and are making the gene product, and this predicts that we'd be able to correct glycogen storage in those muscle cells if we were expressing GAA. And indeed, we published that we could do so in 2008 in a paper by Barang Sung et al. So that's making GAA in the muscle specifically. I also wanted to mention that in this approach, antibodies are irrelevant. With the muscle-specific promoter, GAA is made in those muscle cells. It doesn't have to be taken up from the circulation, so antibodies in the blood really don't matter to this approach to gene therapy. However, we know from several experiments that making GAA everywhere in the body, including the muscle, isn't effective because that provokes T-cell responses. We need the muscle-specific expression to avoid the immune effect in this experiment. That was rather technical. Let's move on. So now, combining the drug with the liver-targeted gene therapy, and so what we've done again is deliver the vector to the liver, expressing GAA there only, but actually a low dose of the vector. And the strategy was to increase the effect of a low dose that might even be considered in a phase 1 trial. Here we can see how the receptor levels would increase at the muscle, plasma membrane, and GAA would increase in lysosomes, and the effect could be improved. So what did we find? Well, actually, it was quite striking after the first 1-month experiment, comparing the rotarod time between mice that received the vector alone, which is our low-dose gene therapy on the left, the gray bars, and then the mice that received the vector plus the drug, the starred bars. So on the left here, you can see how there's about a doubling of the rotarod latency, or rotarod time, which is an improvement in performance that we've never seen before. It was quite striking. But these were comparable groups of mice. There were numbers sufficient to establish statistical significance. So we find this is a compelling result. Also, Clenbuterol by itself, the drug by itself, didn't improve rotarod performance to nearly the same degree. We've looked then at the receptor level in muscle, and these are Western blots. These are individual mice, and this is in A, the extensor digitalis longus, or EDL muscle, comprised mainly of type 2 fibers. You can appreciate perhaps that the first 4 lanes have a slightly stronger signal than the second right 4 lanes. When that's quantified, we can see that there's an increased level of the receptor in the EDL here with the drug and the vector treatment as compared with the vector alone. And we saw actually a similar effect in the brain. And so the large hemispheres of the brain were analyzed in the same way, and in the cerebrum increased CI-MPR by adding Clenbuterol. Just to summarize then, Clenbuterol treatment really increased the efficacy from both ERT and gene therapy in mice with Pompe disease. These were adult immunocompetent mice, so this might predict the response in perhaps a CRIM-negative infant with Pompe disease. Now we saw benefits from the beta-2 agonist treatment, the Clenbuterol treatment, that suggests that this might work as an adjunctive therapy added with ERT. So we propose a clinical trial of the beta-2 agonist drug therapy in patients with late-onset Pompe disease. We'd evaluate safety and efficacy over 12 weeks using standard measures for efficacy. It's very intriguing to look in the literature and see that Angelini and colleagues published the use of one such beta-2 agonist before ERT was available, without ERT, and there was an improvement in the MRC score of muscle function over long-term treatment. So this predicts that such a drug might be beneficial in patients who have residual GAA activity, and we'd say that adding ERT would have a synergistic or improved effect. And here are some of the details. We would do muscle pulmonary function testing, use a secondary endpoint with the biomarker, just three visits to Duke University over 12 weeks. And some of the patients would undergo a muscle biopsy to look at the effect on receptor and GA activity in their muscles. That would be voluntary. So finally, I really wanted to thank all of our collaborators and colleagues at Duke and at other institutions, especially Dr. Rabin, funding sources, Allison McVie-Wiley at Genzyme Corporation, who worked on us with proof of concept experiments, and here's my group. Thank you.